Hi class, we are back in the lab today and we are going to be doing the spore stain today. So before I actually demonstrate this for you, I'm going to go over a little bit about spore formers, the spore cycle, and then talk a little bit about the stain before we actually perform it. So spore formers are bacteria that form spores. And we have to have a special technique to be able to stain the spores because they are so heat and chemical resistant. You can't stain them using the normal gram stain or a simple stain. So we have to have a special stain to stain them. So which bacteria are spore formers? There are two families of spore forming bacteria, and those bacteria are Bacillus and Clostridium. Both of those bacteria are gram-positive rods. Most of those are not pathogenic. There is one pathogenic strain, which is Bacillus anthracis, and that's the bacteria that causes anthrax. And then we have Clostridium, and the bacteria Clostridium, a lot of those are pathogenic bacteria. The three that are mentioned in your lab manual are Clostridium botulinum, which causes botulism, Clostridium perfringens, which is the main bacteria that causes gangrene, and then Clostridium tetani, which is the bacteria that causes tetanus. So those are the three pathogenic strains of Clostridium, main pathogenic strains. So now we're going to talk a little bit about the spore cycle. Now, in lecture, you should go over the spore cycle in great detail, going over each and every step, what's all happening at each step. In lab, that's really not necessary. I'm going to go over 4 main steps of the spore cycle, and the reason why I go over those steps is because you're going to see each of those steps under the microscope. Once you stain the bacteria, you'll see the bacteria actually going through those stages. So I want you to understand what you're looking at when you're looking at it under the microscope. So starting out, we have the vegetative cell. The vegetative cell is when the bacteria is actively living and metabolizing and reproducing. So basically the cell is happy at this point, the environment is viable, pH, temperature, has enough food, nutrients, so everything's good. So that's the vegetative cell. Now, once something stresses that cell out, where pH goes up or down, temperature goes up or down, or it's running out of nutrients, the cell will undergo what is called sporulation. And that's where the spore is formed inside of the cell, the spore coating, and is formed, and then the DNA is copied and placed inside of the spore. So at this time, you can still see the spore on the inside of the vegetative cell. And this is called an endospore because it's still technically inside of the vegetative cell. The next step is once the vegetative cell dies off, you're left with a free spore is what it's called. And this is a lot of times what people think of when they think of a spore. The vegetative cell has already died off and it's just the spore by itself. Now, these spores are dormant. So that means that they're not actively metabolizing and reproducing. They can stay dormant for millions of years if they need to. Then once the environment becomes viable again, they go through what is called germination. And germination is where a new vegetative cell is produced from the spore. So since these spores are resistant resistant to most chemicals and heat, we have to use a special staining technique to be able to stain them. The spore coating is made of what we call DPA, dipicolinic acid, and it's very, very strong. And that's what makes the spore coating so resilient over a long period of time. So, of course, when we're doing these stains, simply applying the stain and letting it set is not going to be enough. So, the stain that we're going to perform today is called the Schaefer Fulton stain, a spore stain. And so what it does is it utilizes heat as the mordant. So the Schaefer Fulton spore stain is a differential stain. So if you can recall back, the gram stain is also a differential stain. So we're going to be using more than one color. The spore stain has those same 4 main steps as the gram stain. It has a primary dye, a mordant, a decolorizer, and a secondary dye or a counter stain. The difference is what each of those are in the spore stain. In the Schaefer Fulton stain, the primary dye is malachite green. The mordant or the fixative is heat. The decolorizer is simply water. and then the counter stain or secondary stain is Safranin. So at the end of it, if it's done correctly, the vegetative cells are pink/red, the Safranin dye that dyes them, and then the spores are the malachite green, which is the primary stain. Okay, so hopefully after I go over the actual staining process, we'll look at it under the microscope and we'll get to see all those different steps of the spore cycle. Okay, so now let's go over and do our stain. Alright, so now we are over here at the workbench and we are going to do our Schaefer Fulton spore stain. I've already got my smear made. As you can see here, I used a bacillus. So we have a bacillus here that we know that forms nice spores. So that's the smear that I use, and I've already heat fixed it and all of that, so it's ready to go. And here is my primary dye, which is the malachite green. Now this is one of the strongest dyes that we use in here, so we really want to be careful. We don't want to get this on our clothes, we don't want to get this on our skin, so I always try to make sure to wear gloves whenever we were dealing with this. So the first step is we are going to flood the slide completely with malachite green, and we're going to let it sit for 60 seconds. So flood the whole, and it's really important to get the whole thing covered because we're about to heat this for 5 minutes. So we don't want our stain to dry up. Now, after we let this sit for 60 seconds, we are going to use our Bunsen burner and we are going to actually flame the stain. This does two different things. Number one, it heats the stain itself, helping the staining power, strengthening the staining power of the malachite green. And then number two, it creates holes in that DPA, that spore coating, so that our stain has somewhere to go. So we're basically cooking this malachite green into the spores. Now, when we do the decolorizing step where we just simply rinse with water, this stain is going to come right out of the vegetative cells because there's really nothing holding it in. But it's going to stay inside of the spores. So after the decolorizing step, if we do it correctly, then our spores will still be green and our vegetative cells will be colorless. So then we're going to counterstain with the Safranin. Now, let me explain the Bunsen burner for you. So the Bunsen burner, okay, you have to use a Bunsen burner to do this. You can't use a Fireboy because we clearly can't pick this up and turn it over. You have to be able to pick it up and turn it over to apply the fire directly to the stain. And now we want to put the fire directly on the stain. If you don't do that, it's not going to heat it enough. But what you don't want to do is sear or cook your smear itself. You don't want to overheat it. If you cook the smear itself instead of the stain, then you're going to crack, your cells will basically crack open and burn, and then you're not going to be able to see anything on the microscope except for scrambled eggs, and we don't want that. So I'm going to light my Bunsen burner, turn my gas on, light my Bunsen burner. Now, I want to turn down the gas a little bit because I don't want that fire to kick back up on my hand and burn me. So I'm going to do that. And of course, I'm going to not pick this up with the hand that I have the glove, wearing the glove, because we, of course, don't want to touch anything hot while we're wearing the glove. So I'm going to pick it up on the bottom, and I'm going to turn it over. And it might spark a little bit, but that's okay. And I'm going to flame the stain. Now you'll notice that I'm putting the flame directly on the stain. That is what I want, okay? I want this stain to steam. We don't want to boil it. We don't want to cook it. We want it to steam. So what I'll do is I'll apply this until it starts to steam. Then I'll let go, let it steam a little bit. Now I want this to steam for 5 straight minutes. After the 5 minutes, I'm going to let it cool for 2 because it's really, really hot before I move on to the next step. And again, this step is the mordant, okay? The fire is the mordant in the spore stain. And after I heat this for 5 minutes, we will move on to the next step, which is going to be the decolorizing step. Alright, so I have heated it for 5 minutes. I've let it cool for 2. So now we are going to rinse it with the decolorizer. So the decolorizer in the spore stain is water. So we're going to just simply take this off. Now this is going to take a little while to rinse because you have just cooked this malachite green onto this slide. And again, you want to rinse till the water runs clear. And so it might take a little while for you to get we want to try to get as much of that malachite green off as we can. And so remember, the malachite green is cooked into the spores. So we can rinse this as much as we want, and it's not going to come out of those spores. So we're going to rinse. All right, that should be good enough. All right, so now we're going to counterstain with our safranin. So we're going to pour safranin, and we're going to let this sit for 60 seconds. Now, after this sits for 60 seconds, then we're going to be ready to rinse again. So we're going to rinse it one last time. Then we'll blot it with our bibulous paper, just like we have the other stains, and then we will be ready to look at it on the microscope to see what we have. Now, one of the things that you want to do when you're looking at these under the microscope is see where the spore is located at. So each bacteria forms its spores in a different location in the vegetative cell. So you've got a centrally located spore, and that is going to be, of course, right in the center of the rod. You have a subterminal spore, which is almost to the end, but not quite. And then you have a terminal spore, which is going to be all the way at the end of the rod. So once we look at ours, we will see where our spores are located at within that vegetative cell. So it's been about a minute, so we're going to go ahead and rinse our safranin off. We'll rinse the bottom too, because we want to make sure that we've got all that stain off of there. And now we are going to blot it dry with the bibulous paper, and then we'll look at it on the microscope. And here is our spore stain under the microscope at 100x with the oil immersion. So the arrow I have pointing at what is called a free spore. So it's that green color, that malachite green that I was telling you about. And this is where the vegetative cell has already died off. Now the arrow is pointing at the endospore, and that is where the vegetative cell has not died off yet, so you can still see the spore inside of the vegetative cell. And you can also, if you look really close, you can see that the location of these spores are subterminal. So whenever I was talking about the three different locations, you have centrally located, subterminal, and terminal. These spores are subterminal, and that helps us to identify what kind of bacillus this is. And then if you have the pink cells that are only pink, then these are going to be the vegetative cells that have not undergone sporulation yet.